Welcome to all of you for today's session of Plant Physiology and Metabolism course. In this lecture, we are going to study the mineral nutrition in plants, particularly uptake, metabolic role, deficiency, symptoms and toxicity of copper, manganese and boron. So, we will take up one by one specific mineral nutrients, that is copper, manganese and boron. You see that copper is like other essential mineral elements. It is one of the micro element nutrient which is necessary for the development of plant growth and development. For healthy development of plant, it generally plant require about 5 to 30 milligram of per kg of copper, and generally this copper is redox active transition element and plays very important roles in like photosynthesis, respiration, metabolism of carbon and nitrogen, and in addition to this, protection against oxidative stress. So you see that most of the functions of copper as plant nutrient is based on enzymatically bound copper, and which catalyzes different reactions. Now, basically, in which form copper is taken up by the plant? So, copper exists in two oxidation states, namely cuprous-ion and cupric-ion. Out of these two forms, the predominant oxidation state found in nature is generally cupric-ion. But in soil, copper is generally found in various forms, including free ions, precipitated forms, exchangeable forms, organic compounds and residual forms. So basically copper tends to accumulate primarily in the topsoil due to its limited mobility. It has a very strong attraction to organic matter, which results in low leaching rates. This is one of the positive aspect which promotes its accumulation in soil. However, what happens, only a fraction of the total copper in soil is actually available to plants. The accessibility of copper to plants depend upon the characteristic of the soil, plant specific factors, and that regulate its mobility and availability. So basically plants predominantly uptake copper in the form of cupric ion and cuprous ion, and the mobility of these ions is generally influenced by different soil properties such as pH and the organic matter content. And copper ions are basically absorbed by the epidermal root cells of the plant and transported through the parenchyma, endodermis before reaching into the xylem. Within the copper transport protein family, you see that including 6 members that have been identified in Rhabdopsis, basically namely COP-T1. This is the copper transport one family, and which is believed to facilitate the copper uptake into cells, and with while other members could also be involved in intracellular transport. And basically these protein family are primarily responsible for the uptake of copper from soil, and generally they have a very high affinity for cuprous ion. Then another protein complex, zinc iron permease, might also participate in the transport of basically divalent cupric ion at the cell membrane alongside another transporter located on the organelle and plasma membrane, which are very selective for both monovalent and divalent forms of copper. Then in addition to this, other type of transporter, namely P type of transporter, which is a heavy metal transporter, move ions like copper, zinc, lead and cadmium, and basically they are also involved in transport of copper. So these are the different protein complexes that have been shown in this slide, COP-T1, ZIP, and these are the protein complexes that are involved in transport of copper. Just have a glimpse of these protein transporter, so you have a very clear view that the type of transporter involved in uptake of copper. Now, basic question is how copper regulate various metabolism inside the cell, and if I talk about at whole plant level. So, like other essential mineral elements, copper basically plays a very essential role, particularly in cell metabolism, by activating various enzymes involved in photosynthesis and respiration. We have gone through in details the various aspects of photosynthesis, and copper is basically also constituent of vital enzymes such as cytochromes, cytochrome oxidase, ascorbic acid oxidase, plastocyanin, polyphenol oxidase. In addition to this, copper is also involved in the receptor of ethylene signal. This is also very important aspect. Copper regulates. Copper also aids in the root metabolism and helps in the utilization of proteins. In addition to this, if I talk about the what is the function of copper at whole plant level. Copper basically provides resistance against plant pathogens. Basically, copper is utilized as one of the fungicides due to its antifungal properties, and copper also serves as a very important constituent of electron transport chain, also in photosynthesis, and also involved in the manufacture of lignin. And in addition to this, it also serves as a component of oxidase enzyme, particularly cytochrome oxidase, and plastocyanin, particularly chloroplast protein. It is also present in root metabolism and helps in the utilization of proteins. So, these are some of the important functions imparted by copper at the cell level and whole plant level. So, now discuss in brief about what are the different copper containing protein. Namely, one of the specific protein is basically copper serves as a what you call that different part of the protein containing copper, and roughly half of the copper present in plant is basically localized in chloroplast, where it binds to plastocyanin and then helping photosynthetic reactions. Another significant form of copper basically include chaperones that binds copper ions and various enzymes, notably oxidases, which contain either one or multiple copper atoms. Copper also plays a very important role in the ethylene receptor and contributes to molybdenum cofactor biosynthesis. Basically, in legumes, you must note at this juncture that a deficiency in copper reduces nodulation and nitrogen fixation. So, basically what happens under copper deficiency, the activity of copper dependent enzymes, the concentration of copper dependent enzyme basically decreases, and often resulting into the metabolic changes and overall hindered plant growth. Then you see that plastocyanin is an integral component of electron transport chain of photosystem 1, and this particular protein has generally, if I talk about what is the number of plastocyanin present, so basically there are about 3 to 4 molecules of plastocyanin per 1000 chlorophyll molecules. So, what happens under copper deficiency? The concentration of copper in leaves correlates basically closely with the concentration of plastocyanin and consequently affects PS1 activity. Whereas the concentration of you see that chlorophyll is minimally affected in case of copper deficiency. But if I talk about how PS2 photosystem 2 is affected by copper, so Of course, there is a subtle impact, or you can say less impact, on the activity of PS2 in case of deficiency of copper. Now, another important copper containing protein is superoxide dismutase. Uh, Basically, this enzyme plays a very important role in detoxification of superoxide radical. That basically this copper atom is generally present in one of the superoxide, copper zinc superoxide dismutase, which is directly involved in the detoxification of superoxide radical, and this radical is basically generated in photosynthesis. So what happens under copper deficiency? Generally the activity of copper zinc superoxide dismutase basically declines in leaves, and basically this decline in copper zinc SOD occurs in chloroplast and cytoplasmic compartments. Now, another copper containing protein is basically cytochrome C oxidase. We have discussed it into detail. So, this cytochrome C oxidase is basically large integral membrane protein which is encoded in the mitochondrial genome, and it is involved in the terminal oxidation as a terminal oxidase of mitochondrial electron transport chain, and generally expressed in the mitochondrial inner membrane. So, you see in the slide that assembly of the basically this active oxidase complex is dependent on the insertion of the 3 copper atom ions along with 2 heme iron and individual zinc, magnesium and sodium ion. So, for proper functioning of cytochrome C oxidase, this copper is playing a very crucial role in its proper function. Now, what is the role of copper in carbohydrate, lipid and nitrogen metabolism? So, you see that as I mentioned that the role of copper in photosystem 1 is very important, and plants lacking copper have slower rates of photosynthesis and make basically less carbohydrates, especially during the early growth stages. So, the lack of carbohydrate in the copper deficient plant basically could result in the impaired pollen formation and fertilization. Basically, this shortage of carbohydrate induced by copper deficiency is could result in that development of nodules, because development of nodule is hampered in case of copper deficiency, and thereby of course nitrogen fixing ability of the plant. But you see that when there is a severe shortage of copper, the plants of the photosystem 2 also change, and the lipid composition changes in favor of less unsaturated fatty acids, and these changes in fatty acid composition in the thylakoids and PS2 complex are probably related to the function of copper in the desaturation of long chain fatty acids. So, these are the very specific functions regulated by copper in the plants, and we should note these crucial aspects regulated by copper. Now, another aspect affected by copper is basically copper deficiency leads to impaired lignification of cell wall, and this is very common anatomical changes noticed in hair plants induced by copper deficiency. And basically this leads to various kind of deformities in young leaves and leads to bending and twisting of stems and branches. So, basically if in case of copper deficiency, it's generally main impact on cell walls, on the cell wall, and you see that how it's going to affect the lignification in the cell wall. So, you see that if there is an excess of copper in if there is excess of other substrate like alpha cellulose and hemicellulose, this could lead to particularly noticeable changes in cell wall, and in plants that is that are severely deficient in copper. The xylem vessels are basically also inadequately lignified or not properly lignified. So, even in case of mild deficiency of copper could lead to reduced lignification of cell walls, that serves as one of the important indicator of nutritional status of copper inside the plant. Now, what are the specific deficiency symptoms induced by copper that can be noticed visually? So, you see that particularly plants growing in soils with low copper content such as ferritic and ferruginous coarse textured soils or calcareous soils derived from chalk and could occur in such kind of soil. And copper deficiency can also occur in soils rich in organic matter, and generally in such soil copper is generally bound to organic substances, therefore not available to the plants. So, high nitrogen availability can exacerbate copper deficiency in plant or could trigger the copper deficiency in the plant. Different plant species actually vary in their sensitivity to copper deficiency. Like you see that wheat, oats and spinach, they are very sensitive, but you see that pea, rye and oilseed rape, they are very they are not very sensitive to copper deficiency. But in case of deficiency, generally copper deficiency could lead to stunted growth, distortion of young leaves, chlorosis or necrosis starting at the tips of the leaf you see in the slide, and then these extend along the margins and leads to bleaching of young leaves, particularly known as white tip or reclamation disease in cereal, particularly grown in organic soils. And it could also induce a very important disease, summer dieback disease in trees. And copper deficiency could also lead to basically secondary symptoms like enhanced formation of tillers in cereals and auxiliary shoots in dicotyledons, resulting from necrosis of the apical meristem. In addition to this, wilting in young leaves is generally observed in copper deficient plants, which could occur due to the impaired water transport resulting from the insufficient lignification of the xylem vessels. In some cases, copper deficiency could also lead to exanthema disease. Exanthema disease basically is the formation of deep slits in the bark from where gums generally exudes, and this disease is popularly induced by copper deficiency known as exanthema disease. Now, opposite to the deficiency, what are the specific toxicity symptoms? If I talk about excessive amount of copper could lead to various kind of adverse effect in the plant, and basically various copper based fungicide used in the agriculture, particularly in vineyard, could lead to various kind of toxicity symptoms. And you see that basically the higher concentration of copper could lead to impaired root growth, and basically it could also trigger the deficiency of other essential mineral element like iron. So toxicity basically induces the deficiency of other essential mineral elements, particularly copper toxicity could lead to iron deficiency in the plant. Now, another mineral element is manganese, which is also one of the essential elements found in all living organisms, particularly in plants. It acts as an enzyme cofactor or as a metal with catalytic activity in various biological clusters. However, just I mentioned the case of copper, its excess could be detrimental for the plant health. So let's discuss about its role in plant and how in which form this manganese is taken up in the plant, its crucial role and metabolism, and then deficiency and toxic symptoms of manganese. So basically, manganese can be found in various oxidation state. That is, in manganese could be found in first, second, third, fourth, and six and seven oxidation state. However, in biological system, generally these oxidation state of 2, 3rd, and 4 are very common, and among these manganese 2nd and manganese 4 uh, are quite stable, while manganese 3rd is less stable. And within soil, generally manganese exists as insoluble oxides or in chelated forms. Once uh, manganese is released from chelates or reduction in higher valence oxide, it is primarily absorbed as the divalent manganese cation, that is Mn2. Roots basically very quickly uptake manganese ions, particularly manganese cations, which are then efficiently transported to shoots. And the movement of manganese ions from root to shoot is very rapid, due to which uh, manganese is very less toxic to roots as compared to other metals present in the soil. And in addition to this, manganese can also undergo oxidation to manganese 3rd and manganese 4, and showing a very crucial role of manganese in redox processes. Now, like other essential mineral elements, this manganese also impart its very specific role in cell metabolism and at whole plant level. Basically, this manganese serves as a catalyst in the growth processes, and it's very important constituent of oxygen evolving complex, which is responsible for the splitting of water during photosynthesis. And in addition to this, manganese also acts as an activator for various enzymes such as maleic dehydrogenase, oxalosuccinic dehydrogenase, and nitrite reductase, like this. In addition to this, manganese plays a role in oxidation of auxins and is essential for the chloroplast development. So this is also a very important function of manganese, that it is playing a role in the oxidation of auxin and is essential for development of chloroplast. And basically, one of the form, that is manganese 2, is required for the activation of numerous enzymes involved in photosynthesis, respiration, and metabolism of nitrogen. It also acts as an electron donor for the chlorophyll B and participate in decarboxylation reaction during the process of respiration. And you see, these are the specific roles played by manganese at various level of cell. But if I talk about what is the role of manganese at whole plant level, so basically manganese accelerates seed germination and maturity, and making availability of or you can say enhance the availability of phosphate and calcium, and that's why contributing overall plant health and vigor. Now, this is one example where manganese is playing a role as a constituent of manganese SOD, manganese superoxide dismutase, and basically this superoxide dismutase are present in all aerobic organisms and play a very essential role in the survival of organisms in the presence of oxygen. And basically these SODs protect tissues from the deleterious effect of oxygen radical, superoxide radical formed in various enzymatic reaction. The conversion of basically superoxide radical is catalyzed by superoxide dismutase, and this leads to formation of, you see in the reaction, this lead to the formation of you see that this leads to the formation of hydrogen peroxide and oxygen, and then hydrogen peroxide is further broken down into water and oxygen. This could be catalyzed by either peroxidase or catalase. So this is one is very important reaction of catalyzed by superoxide dismutase. Now there is another important well-known extensively studied case of manganese containing enzyme is the polypeptide found in the water splitting system in PS2. Within this mechanism, you see that a cluster containing comprising 4 manganese atoms accompanied by 1 calcium atom and forming manganese 4 calcium is organized uh, and facilitating the 4 electron oxidation of 2 water molecules. So you see that how manganese containing SOD is playing a role in water splitting system associated with PS2. Now, role of manganese in tricarboxylic acid cycle. Basically, this manganese functions as a cofactor and basically activating a number of enzymes. This could be around 35 distinct enzymes that are regulated by manganese. So majority of these enzymes are involved in catalyzing oxidation reduction, decarboxylation, hydrolytic reactions, and manganese basically has a very primary role in tricarboxylic acid cycle in oxidative and non-oxidative decarboxylation. For example, NADPH specific decarboxylating malate dehydrogenase, malic enzyme, and isocitrate dehydrogenase. For example, this manganese is essential in the enzymes such as NADPH specific decarboxylating malate dehydrogenase, malic enzyme, and isocitrate dehydrogenase. So these are the some very specific enzymes where involved in tricarboxylic acid cycle, where manganese is playing a very crucial role. Manganese is playing a very crucial role in the bundle sheath chloroplast of some of the C4 plants, where you see that oxaloacetate serves as the carbon shuttle, and decarboxylation is basically facilitated by phosphoenol pyruvate carboxykinase. So the enzyme basically phosphoenol pyruvate carboxykinase basically relies on the manganese that cannot be replaced by magnesium cation. Manganese deficiency generally encountered in plants, particularly growing in soil derived from low manganese as well as heavily leached tropical soils. Generally, manganese deficiency is common in soils with generally high pH levels that contain free carbonates, especially when combined with high organic matter content. So you see that in slide this manganese deficiency generally causes a disease known as gray speck disease in oat, and in addition to this manganese deficiency also leads to symptoms in the cotyledons as known as marsh spot, particularly in peas, and split seed disorder in lupins. You see that interveinal chlorosis with gray spots on young leaves is very common symptom of manganese deficiency, and generally leaf appeared very much malformed, and generally streaks appear on the leaves of some plant. And overall, if I talk about what is the status of growth in case of manganese deficiency, its plant generally have a slow growth. Basically, in manganese deficient plant, generally accumulation of dry matter, net photosynthetic rate, and chlorophyll content decline very rapidly, and generally manganese deficient plants are more susceptible to damage by low temperature, freezing temperature, and generally very much prone to be affected by various root rotting fungal infections. And overall, what is the impact of manganese deficiency? That generally it hampers the uh, grain grain yield, in plant, and generally it also affects the pollen fertility due to short supply of carbohydrate during the grain filling stage. So manganese deficiency is going to affect basically overall yield of the plant. Now I talk about the toxicity. First, what are the specific toxicity symptoms induced by manganese? Basically, manganese toxicity induces brown speckle disease on mature leaves. You see in the slide. The intensity of basically this brown speckle formation is generally used as indicator of manganese level in the soil as well as its tolerance level. And this manganese toxicity could also lead to intraveinal chlorosis and necrosis. And generally, this toxicity symptom induced by distortion in young leaves known as crinkle leaf, which is a characteristic indication of calcium deficiency. Then manganese toxicity could lead to induced deficiencies of other nutrients like calcium, manganese, iron, and zinc. So the induced deficiency of iron and magnesium results from inhibited uptake across the plasma membrane and competition, or you can say imbalance at the cellular levels. Additionally, manganese toxicity could also lead to loss of apical dominance and increased formation of auxiliary shoots, commonly referred as witch's broom.